Good afternoon or good evening. I am very pleased to introduce the next workshop, which is SPEAQeasy with Nicholas, Leo, and Josh.
Thank you for coming. So I'm going to share my screen and just show you how you can follow the workshop using the Docker images that we've created and an app that Sean Davis created for us with the help of Ken Brew Albrecht. So I'll share my entire screen. So if you go to the Eurobias C2020 website, you'll see on the left side here that says workshop. When you click on that, you'll notice that we have a cloud platform. And so I'm going to open this website, which is app.orchestra.cancerdataSci.org. And so you can find all the different workshops here. So under the search bar, I'll search for SpeakeAsy, which is the name of the software that we're going to teach. And then you click on the blue button on the right that says launch. That will launch a specific instance for you. Each of us will have a different URL. For now, it's loading these 8 SL, etc. for me. I already had one loaded because you can see here that it lasts up to 8 hours. So I'm going to go to my tab where I already have it loaded. And what you'll see here is a regular RStudio window. Now, on the bottom right, you'll find the files, plots, packages, help, and viewer navigation. If you click under help, you can go to reference where it says packages on the bottom right. Once there, I will navigate near the end because our package name is SpeakeAsy Workshop 2020, which I passed. SpeakeAsy Workshop 2020. Under that, I can see the package vignettes, etc. So I'll click there, and I'm going to open the HTML version of it. Why did I do all of this? That's because now you can see the render version in nice HTML format of what we're going to show you today. But you can also, for example, highlight some code and press Ctrl-Enter on your keyboard, and that will automatically evaluate the code on the console terminal on the left side. So, for example, here I'll run some R code later on. You can see that. So that way you don't have to copy paste any of the code, and it's going to make it easier for you to follow what we're teaching. So with that, I'm going to let Nick explain most of the SpeakeAsy. This workshop was going to be taught by Nick and Josh, but I'll be doing mostly what Josh's part because of he's not feeling great. Although Josh is still around today, in case you have any specific questions for him.
Thanks so much, Leo, and thanks for the hosts for allowing me to speak at this conference. Okay, yeah, as Leo mentioned, I'll be presenting along with Leo a pipeline called SpeakeAsy, which stands for a Scalable Pipeline for Expression Analysis and Quantification for R and Bioconductor-powered RNA-seq analyses. And originally, it would be me and Josh presenting. We have the GitHub links. So just as a brief introduction for what SpeakeAsy is before we start actually interacting with it, I provided some links here, and I recommend that you follow along with the slides, which are uploaded to the location, F1000 location. And so I guess the materials for this workshop are going to be the vignette with all the code, the SpeakeAsy repository itself. There's a documentation website as well for SpeakeAsy, and then also we've used this pipeline so far in 2 of our papers. Yeah, so what is SpeakeAsy? It's a scalable pipeline for expression and quantification. So basically, it's an RNA-seq computational processing pipeline that's built with Nextflow. So another common tool used to build pipelines is SnakeMake that you may be familiar with. but we built ours using Nextflow. The emphasis here for SPEAQeasy was really making it easy to install, use, and share among researchers. So really, it takes raw FASTQ files from sequencing, and the main products of interest are summarized experiment objects. So really a backbone for a lot of bioconductor analyses. And these summarized experiment objects contain 4 different features. So genes, exons, exon-exon junctions, and transcripts. So like having all the data collected into a summarized experiment allows you to really integrate it right away with a number of bioconductor packages, depending on what you're trying to do. It really allows you to guide the analysis in the direction you want using your favorite package. Okay, so just to provide an overview of how the pipeline goes, I know on the right it looks like a big complicated workflow, but I'll try to summarize it more clearly. So starting with the input FASTQ files from sequencing, we do a quality check with FASTQC, trimming, and I should note that the yellow processes on the right are optional steps or steps that aren't always run. So trimming is very configurable with SPEAQeasy. By default, it's quality and adapter trimming based on whether the adapter content metric from FASTQC passed. But you can customize it really to do any type of trimming, so quality or adapter trimming, or both, or just not trim at all. We also have a step to automatically infer and confirm the strandness orientation of the reads. So like the strandness is something that would be a consequence of the library prep. And some computational tools like Callisto, for example, will require you to provide the strandness. And so that's kind of why we check. And SPEAQeasy can just confirm that the strandness is as expected. So if you're not familiar, like sometimes the strandnesses are called like reverse, forward, or unstranded. There's also support for experiments with ERCC spike-ins as a quality control. So this pipeline performs both alignment to a reference genome and also a pseudo alignment to a reference transcriptome. And then I guess the other big thing is that for human samples, there's genotype calling, and we'll really get into the details of that later. So ultimately, that can be used to create a genotype, a unique genotype profile for each sample, and then you can use that to resolve identity issues. But we'll get into the specifics of that later in the workshop. So there are a good number of publicly available RNA-seq pipelines already out there. So I'm just going to get into why you might be interested in SPEAQeasy over the ones that are already available. So one of the things is the annotation. SPEAQeasy has a very automatic method for managing annotation in reference files. So we've seen in a number of other pipelines that typically they expect you to provide your own files for doing this. That is an option with SPEAQeasy, but there's also the default more automated method where in the configuration you specify a GenCode release for if you're doing experiments with human or mouse organisms, and an ensemble release for rat. And the other variable that you can control with the configuration is choosing between what we call a main or a primary genome build. And that's designed to follow the nomenclature that GenCode uses. So like GenCode, one of the like reference files you can get from GenCode is called like the primary assembly build. And that includes the canonical reference sequences. So like the first chromosomes 1 through 22, X, Y, and the mitochondrial chromosome, but also additional scaffolds or alternate chromosomes. And if you don't want those additional scaffolds and alternate chromosomes, you select the main build, which only includes the canonical reference sequences. And basically, like there have been some papers showing that keeping annotation consistent is important. And so SPEAQeasy tries to really keep that consistent throughout the whole pipeline. And in terms of automation, so the specified files are automatically downloaded on the first run. And then if you choose the same settings on a future run, it just takes the cached files. So it's not like it has to re-download everything. But of course, if you want to circumvent this and you want to provide specific files yourself, that's an option. You can just specify your own. Another thing we emphasized with the speakeasy pipeline was that it should be pre-configured for a lot of computational environments. So a lot of pipelines that we've seen have, in theory, support a lot of different platforms, but in practice, configuration is really left up to the user for a number of platforms. And we wanted to just have it kind of work out of the box for common environments. So specifically what we mean is that so computing clusters managed by the slurm or SGE resource managers, we wanted speakeasy to be able to work on those out of the box. You can also run it on a local machine, and it supports execution with Docker containers, which I'll get into a little bit later. We also have the feature for quickly and automatically determining the strandness of samples as a quality check. And one of the big things that we again, we'll get into the details of this later, but for human samples, like I mentioned, there's variant calling is done to produce a unique genotype profile. You can compare with the pre-sequencing genotyping data if you've collected that, and ultimately that can be used to resolve identity issues. We sometimes call that sample unswapping. And this little graphic at the bottom shows a self-correlation heat map. So this is sort of correlations produced by assigning each genotype a numeric value and computing the correlation between samples and themselves. So that's why the diagonal has a correlation of 1. And then immediately you see that in this example, sort of the orange spots that are off the diagonal and the red off the diagonal points to identity issues. So samples shouldn't be correlating with samples of a different identity, basically. And we'll get into the specifics of this later in the workshop. A couple other things. So we mentioned ease of installation. So we wanted to be able to have speakeasy installed in a single command and not having to worry about root permissions or just what dependency management tools you have installed, like Anaconda is a common one. We wanted to make it just super simple and something you can install in a single command. Another feature is being able to automatically merge samples that are split across multiple FASTQ files. So that might happen if you're sequencing with multiple lanes and you want to associate several FASTQ files with a single sample. And to get more concrete about how you do that, the inputs to the pipeline, actually it's a single input. You just specify the path to a file called samples.manifest. This is the example here is for paired end samples. And we use a format that's sort of designed to have backwards compatibility with this tool called Rail RNA. So in that tool, for paired end samples, you specify the path to the first read, an MD5 sum for the first read, path to the second read, MD5 sum for that, and then the identity or an identifier that you want to associate with the sample. In speakeasy, we don't actually verify file integrity with MD5. So in practice, we typically just put zeros. So that's what you see in this example. And getting back to the file merging, you see in this example, the first 2 lines have the same sample identifier at the end. So that signals for speakeasy to combine them and treat all of those files as if they have the same sample identity. So that's a convenient way to automatically merge things. And to get more detailed into the speakeasy outputs, these are, again, range summarized experiment R objects. They're objects for both the raw and the normalized counts. And in the call data slot of these objects, they are quality metrics collected from different steps in the pipeline. And we collect information about 4 different feature types, which are genes, exon junctions, and then transcripts. And the quality metrics collected include things like metrics from FastQC, from trimming, from alignment, and if applicable, ERCC pseudo-alignment. So when for human samples, the genotyping is done and produces a single VCF file for all the variants, or for all samples. And an optional output is our big coverage files that are done per strand, and also the mean coverage across both strands. And another step we do using the DirFinder package is computing expressed regions in the form of an RDA file. And we keep this optional because it's a somewhat specific step. It's a precursor to computing differentially expressed regions, and that's if that's of interest for your analyses. But it's optional since that's not always the use case. Okay, so next we'll get more hands-on into the actual pipeline. So just to mention again some of the links, I definitely encourage you to follow along with the vignette Leo also mentioned the Docker image that you can use. The vignette also has this command and all the details, but I think he touched on that in the beginning. Speakeasy repository, which you can clone using that link. And also the R package contains some example files included as an alternative to the Docker image. And you can install that with remotes install _GitHub. Also, BioC manager install also works instead of remotes install GitHub. While everyone gets started with that stuff, I can just talk about, so if you are trying to set up a pipeline normally, this isn't going to be required for the workshop because depending on the way installation method you choose, it can take a while. But this is just to go into the details, just to get familiar with them. So you would change directory into the Speakeasy repository, and then you choose how you want to manage your dependencies. So we recommend doing that with Docker if it's available. And so the way you would install the pipeline once you've cloned it is to just run the script this bash script we have. So that'd be bash install _software.sh and Docker as a string if you're going to manage dependencies with Docker. So we wanted to have a way to install things without tools like Docker, just as an option, just to keep things super simple. But we do recommend Docker if it's available. The alternative is building dependencies from source with our scripts and just passing local to that script instead of Docker. And that will take a lot longer since building things from source can take some time. And that's it. That's all you have to do to install the pipeline. Next, to get ready to run it, you would select what we call the main script and an associated config file. The main script is the script that you would actually submit as a job or run interactively to run the pipeline. So that depends on your computational environment. So if you have a cluster managed by the slurm scheduler, you would choose run _pipeline _slurm.sh just in the main directory. And then the associated config would be in the conf directory, docker _slurm.config. And note, if you didn't use Docker to install, it would be slurm.config instead of docker _slurm.config. But the naming convention is the same for if you use a SGE managed cluster, if you're running it locally, or at the Johns Hopkins Performance Computing environment, where we developed Speakeasy. But yeah, so those are the files to keep in mind. And just to make things a little more interactive, we have an exercise just to explore sort of what you can configure with Speakeasy. And I encourage people to try this out. Let's say you wanted to use Speakeasy on a Slurm managed cluster and you did have access to Docker and wanted to do that. So see if you can find how to adjust the configuration for the trimming process to give it more hardware resources. So in this case, let's say 5 CPUs and 20 gigabytes of memory. So I haven't mentioned specifically how to control memory, but hopefully if you open up configuration, Nextflow makes it really straightforward to specify these things. So I do encourage you guys to try this out. And a hint would be that like in the appropriate config file that you choose, this will be in the process section. So there's a few different sections. I think params is one of them, executor is another, but this will be in the process section. And then a second thing to try out is seeing if you can adjust the configuration to use the latest Gencode release for the human reference if you're doing that. So I think that's still 36 if I'm not wrong. And I encourage people to try this out for I think maybe 3 to 5 minutes or so. And feel free to if you run into anything or have any questions, you can use the questions.
There isn't any questions at the moment.
Okay. Yeah, I'll just give it a few more minutes, and I'll also just go through the solution if you want to wait for that. But I'll give it a couple minutes.
If you want, we could do a quick poll to see if people need more time to do the exercise. Or
yeah, actually, that'd be great. That'd be great.
You want me to do a quick poll on that so you get a better idea?
Yeah, that'd be useful. And just by the way, if you're following along with the slides, we do include links to things like the config, so that if you online instead of cloning the repository and stuff, that's an option.
There's 75% are done.
Mm-hmm. Okay. Yeah, I think I can just continue. If you tried that out, so first my hint was finding the process section in this config. So first of all, I opened up. But yeah, okay, I did my own exercise wrong, but I was supposed to open docker_slurm.config. It should look very similar. So here's the process section, as I mentioned. There's a lot of different things you can specify, but we were looking for the trimming section. So, okay, so in this case, we see that the defaults were 4 CPUs and 16 gigs of memory. So to do the exercise, I can just simply change this to 5 and 20. And that's really the same thing you would do for any other process. Some of the names are a little bit more harder to infer, but okay, well, that's the first part of the exercise. The second part was we were looking to adjust the GenCode version. And that actually is right at the top here. So it's right now it's set to 32, but the most recent is 36. So I can just change it to 36, save it, and then when we run speakeasy, those things will be updated. Okay, and I just wanted to go into like what you would change in the main script when you're running speakeasy and your personal data. So like the main 3 options that change based on the experiment are the reference genome, which can be, we support human, mouse, and rat organisms. So the build specifically HT19, HT38, MM10, and RN6. So those are the options you can pass to the reference argument. You specify whether your samples are paired end or single end. That you pass that to the sample argument. Also, the expected strandness pattern that you have for all your samples, one experiment should have a single strandness pattern. So in speakeasy, those are called forward, reverse, and unstranded. Note that forward is sometimes called like FR stranded for paired reads. Reverse is sometimes called RF stranded. There are a bunch of different names, but those are the 3 options. So to get more into the details of like, let's say we're running this on for a particular experiment. So this is what the full main script would look like. So this should be, in this example, I'm using run_pipeline_slurm.sh. And there's a lot of stuff, but the stuff that you would be adjusting is this middle section. So like lines 13, if you can see this, lines 13 through 22. So we see the main.nf command. That's like what runs the pipeline. The sample argument, this would be for paired end reads. We're specifying that we want HT38 as a reference genome, and our strandness we expect to be reverse. The -w argument here is an argument that I recommend you use, especially if you are planning to share the pipeline with other users. So it's the working directory so for temporary files that speakeasy will generate. The reason I recommend this when you're sharing the pipeline is because if you share a working directory with other people, well, the working directories get very large, and in practice, you probably will want to be able to delete them to free up space for large experiments. And if you do that while someone else is running their pipeline, it interferes with their execution. So I always recommend keeping that to a directory that you personally own. The --input argument is this is a bit confusing, but it's the directory containing the samples.manifest file that I mentioned earlier. The --output argument is just the directory of outputs for where speakeasy will put the outputs. --annotation you don't have to worry too much about. You can just keep it. If you keep it as its default setting, that should work for sharing the pipeline. So sharing the pipeline, you would copy this main script, and based on the way this is set up, it will use the same annotation directory that as like in the original installation location. So like if multiple users use the same annotation settings, they can benefit from like if one person runs it, then it's cached for everybody else. That's the idea. So in practice, you shouldn't really have to modify this argument, but that's what it's doing. An optional argument here is the with report. That's something provided by Nextflow. And it's it might interest you. It's a more visual HTML document of when the pipeline completes, like how much memory was used for each process, which things succeeded. Hopefully everything should succeed. And just other details like that might interest you. The one argument that you is important to change potentially is the dash profile argument. In this case, the dash profile argument points you to which config you're using. So if you're using the slurm.config, it's correct. But if you're using the Docker slurm config, you'd have to change this argument to dash profile docker underscore slurm. And the same goes for if you're using like SGE as well, you would change it to docker underscore SGE. But yeah, that's that. And to actually run the pipeline, Speakeasy is really intended to be submitted as a job if you have access to a cluster that manages that. So like, in other words, if you're using Slurm or SGE, you should submit Speakeasy as a job using the command appropriate for whichever one you're using. If you're running like locally, you can just run it interactively by executing the bash scripts directly. So the sample commands are given below. So like Slurm users would use the sbatch command. SGE would use qsub. So once you submit it, how do you know when it's going to be done? You would check the log called speakeasy underscore output.log. So that's a log provided by Nextflow, and a lot of it is very specific to Nextflow, and it's hard to interpret, and it can be a very large file. But the one thing it's very useful for immediately is that it indicates whether the pipeline completed at the bottom. Speakeasy actually generates its own logs that can be useful for other reasons, but just to check whether it completed, you should check the speakeasy underscore output.log. Okay, so in this next section, I'm going to go through the vignette. So just to take a look at the Speakeasy, the summarized experiment output objects from Speakeasy, just to get familiar with what they look like. So Leo mentioned the option with the Docker container, but I didn't get that set up. I'm just going to run this in my RStudio. And I'm going to first, so I already loaded the Speakeasy workshop package. That'd be the first step here.
Nick.
Yeah.
We have a question that says, can you do a drive run of the pipeline to make sure all the options are inputted are right? So you click on their session on the top right of your screen. And then Q&A on the middle. That's where you can see the question. Yeah.
Can you do a dry run of the pipeline to make sure all the options you inputted are right? Okay, so this is for input. I don't believe we provided like input data for the run. So this was just an example of this is actually from the Speakeasy example. Which so like the options.
I think the question might be more in general. Like, can you, for example, just run Nextflow without actually
okay,
having all the jobs submitted, right?
Yes, I just to test that like it's installed. Okay. I mean, you should still be able to submit.
So like, this would be like a Nextflow feature, right? Whether Nextflow allows dry runs or not.
Yeah, if you're just testing that like Nextflow is installed correctly, I think I'd have to check the documentation for that. But I think it does. Nextflow has like a command for that. So like, I think it's just like Nextflow space like something. I'm sorry, that's not a great answer. But for Speakeasy, you should be able to just try the commands and the command given here to submit the pipeline. And even if things aren't like installed, it should physically run the pipeline, and it would error out on like a software tool that wasn't installed. I guess that would be one way to test that it's like things are set up as you expect, but not yet installed. There's a question. But yeah, I'm going to move on to this section. I'm going to look at the vignette, which we can use to take a look at the example output. So this is in the get started section. I already loaded the R package. So the next thing I'm going to do is, find the section. So in this section, we load the example files. I'm just going to copy and paste the command for that. Okay. And you guys can do this too. The next section would be, I'm going to load some packages that we'll use to examine these outputs. So for summarize experiment, of course, since that's what we're what these objects are. Also, this package Chaff Lab, which has a utility that we'll use. And once those things are loaded, the next step would be to load the example RSE, the arranged summarized experiment objects that's provided in this R package. So I'm going to go ahead and load that. Okay, and so it loaded this object called RSE gene. So that this is the arranged summarized experiment just for the gene features. So there's also one for exons, exon junctions, and transcripts. But okay, so first step, we can just see the default summarized experiment output that is given when we just print the objects. Okay, so this is a range summarized experiment. We would look at the dimension. We can see that it has 60,000-ish rows. These rows are genes, correspond to genes. 42 columns, so that's 42 samples. It has one assay, which is the counts, in this case, the raw counts. The row names would be the gene names. Row data is annotation for the genes. And we'll get more into the call data. So let me just follow along with this vignette. So we mentioned that Speakeasy collects metrics throughout the pipeline for each sample, and it puts that in the call data slot of the RSE object. And if we look at the call names of that, that would give us the names of the metrics that Speakeasy collects. So it's actually a lot of them, but without going into them individually, at the top you can see some metrics collected from FastQC. So like FastQC basic stats, per base content, adapter content, and stuff like that. It keeps a metric as to which samples are trimmed, since by default trimming is done based on the adapter content of the sample. Not all samples are necessarily trimmed. Some stats from alignment, so like overall map rate, so total assigned gene, and like the rate of reads mapped concordantly, and details like that can be useful covariates to control for. Also, another interesting one is rRNA rate. So like the percent of reads that map to ribosomal RNA. So depending on your library prep, you might expect that to be extremely low. And that can be something that you can check. Okay, so we've looked at that. Let's just take a look at what the counts physically look like. So that you would do that with the assays, the counts assay specifically. And then this corner function is from the Chaff Lab package. It's it basically takes like the top left corner of a matrix or a data frame, which can be convenient if there's a lot of columns. So that's how it differs from head. So we see that certain genes have like 0 counts, but other ones have quite a few. And this is just the top left corner of the matrix of the data frame, I guess. Okay, so next thing we can examine is the row data. So the row data is the, in this case, data associated with the rows would be gene annotation. And we're just going to take the top of that just to see what kinds of annotation there is for each gene. So we have like a gen code ID for the gene, ensemble ID, the type of the gene, and all this, most of this information is collected from the annotation GTF, which is automatically like managed by Speakeasy from gen code in this case, since we're this is an example with human samples. We have the gene symbol. Another thing that we collect is the Entourage ID, so that can be useful for gene ontology analysis. Unfortunately, not all genes will have an Entourage ID, so you see the top ones are NA, but that can be useful to collect. And then we have things like mean expression as well. And that's just like a good, like a brief overview of like what the object looks like. Okay, so the next interactive segment of an exercise, just to get familiar with these speakeasy outputs. So see if you can try to find samples in this example that I showed, samples which passed the SPEAQeasy adapter content metric and also had more than 2% of the reads aligned to the mitochondrial chromosome. And you'll need to know the names of the variables, and those variables are called adapter content and mito rate, respectively. So I'll give that a few minutes, see if you can try to load those example objects and see if you can try this out. Maybe we can do a similar poll type thing as from the last exercise.
I've started the poll.
All right, thanks.
It's just simply, do you need more time? Yes or no?
Yeah, that works. Okay, let's see.
Okay, the early vote is 100% no. Give 5 seconds for people to come in with a yes if they want. But
Yep.
if not, I think we might be able to proceed. There isn't any quick questions in the Q&A at the moment.
Okay. All right. Yeah. I can just continue then. Let me do this quickly. Okay. So we're looking for a couple different metrics for these samples, and a particular metric corresponds to a column in the call data slot of the RSE gene object, and we can use the shorthand $sign notation for accessing columns in the call data. So just to form like a vector of the indices that we're interested in. So again, we're looking for past the SPEAQeasy adapter content metric and 2% or more reads aligned to the mitochondrial chromosome. So that's the adapter content variable. If you check that out, you can see that there's either pass or fail. So we want pass. And then the mito rate variable, we want to be greater than 0.02. So that's a logical vector. And to get that logical vector aligns with the sample names. So it lines up with the sample names. So to get the actual sample names themselves, we would take the call names and then use that logical vector to subset. And if you did that, you should get 7 samples that had those criteria met. So that's the end of the initial segment. And in the next segment, we'll be doing an example differential expression analysis given the summarized experiment outputs from speakeasy. And as we mentioned, this had been led really by Josh Stoles, but he's not feeling well. He'll be in the chat, I believe, to answer any questions, but otherwise this will be led by Leo for now. And I'll just go ahead and hand this off to Leo.
How's everyone doing? Thank you, Nick. There's a question about, can you share the slides after? They seem very useful. So I know you mentioned that the slides are on the F1000 website. Can you just post the link?
Oh yeah.
So like I mentioned earlier today, this part might be easier if you open the workshop from the cloud platform on the EuroBioC 2020 website under workshops. If you click this link, there you can find the Speakeasy workshop, lunch. Once you click lunch, you'll have a little website like this, where if you go to the help panel, under reference packages here at the bottom, if I click on packages, once it loads, you can scroll and find Speakeasy Workshop 2020. Click under user guides, package vignettes. I'll click here under HTML. And so at this point now we have what I rearranged my window a little bit. So I have on the left the console, the art console. on the right, I only have this HTML window with a vignette. And I'm going to do this because it's going to make it easier for us to select some lines of code, for example, here, and then press Ctrl-Enter to run the code on the console. So this is if you want to run the code yourself live, but you can otherwise just follow on the vignette. So we made SPEAQeasy because we had quite a bit of bulk RNA-seq data at the Liber Institute that we were working with, but we're also R and Bioconductor users, so we wanted to have easy to analyze R objects, right? And so that's why we're sharing these files as range summarized experiment objects from the summarized experiment package from Bioconductor. And so Nick has shown you a little bit about this object over here that was created by SPEAQeasy. I forgot to load it. So once we load it, we can explore it a little bit. So here we have the data. The gene counts for 60,000 genes across 42 samples that are part of this example data set. And we have several quality metrics that were produced from SPEAQeasy, such as the rRNA rate. Now, that's not the only type of file that SPEAQeasy produces. There's also information here that we can use for identifying sample swaps. So how can we identify sample swaps? Well, SPEAQeasy generates, quantifies variants, coding variants. So these are variants that you can observe in RNA-seq data that are common across individuals, and that you might have also generated data for the same samples using a DNA genotyping assay. So if you have the RNA infer genotypes and then the DNA genotypes, you can try to find if there are any matches or not. And if there are matches, which you should expect to have matches, if the labels for the RNA and the DNA are the same labels. If they're not the same labels, let's say brain number 1 from the RNA matches to brain number 5 from the DNA side, then you might have a sample swap. You can try to resolve it. So this is work that was written initially by Josh Stolz and Luis Huki. And so I'm going to load here a couple of packages that we can use for the sample swapping. So one of them is the heat map. So we're going to make some heat maps. tidyr, because we're going to use some functions here for manipulating data. And then variant annotation, which is a Bioconductor package that allows us to work with what's called a VCF file, or a variant common format file. As part of the set of examples files, we have this hide button is getting on the way, but we have a little file here that I want to load. That is the DNA genotyping data for the same set of brains that we used from the RNA-seq side. We have also the phenotype data for those brains. So this is our DNA information that is external to SPEAQeasy. Now we get into the SPEAQeasy outputs, and we can look here at we have this merge variants VCF file that was produced by SPEAQeasy. We have it here on the pipeline outputs directory. So let's read it in using functions from the variant annotation package. So I accidentally pressed it twice. And we have here also some information about our brains. So now we have all this information here. We can try to see if there are any swaps. And so one thing that we can do is we can look at the genotyping information. And so using the geno function from variant annotation, we can look at our speakeasy object, which is the one that has the the variant common format file ready. Sorry. And we're going to look at the genotype here on the GT. And when we look at that table there, we see that we have all these genotypes here for these variants, which is we either have a 01, 02, 11, 22 for the different alleles. The ones that are dot / dot, those are we're not able to be accurately determined, so we can ignore those. We're going to turn them into NAs. And so let's work a little bit with this data. So
Lou, can I ask, there's a couple of questions here. So
sure.
Devika wants to know, are there detailed instructions to do the variant analysis on your own samples?
What do you mean by the variant analysis? For generating the DNA genotyping data? Is that your question, Devika?
If the person who asked the question wants to raise their hand, I can.
Yeah, we can promote you so we can speak.
Promote you if you want to have a discussion on this. I'm not sure. I had one question myself, which is, can you do variant analysis on RNA?
So you can only a small fraction of the variants are coding variants. So that means that they overlap genes, and then are thus measured by your RNA-seq data. Something that you have to take into account when you're working with coding variants from RNA-seq is that you might not have enough reads across your variant, right? So let's say you have a single read that overlaps your variant, and maybe that read tells you that instead of the more common genotype, let's say A, you have a T. You might not believe that because it's a single read, but you have, let's say, 5 or 10 reads, then you might start to believe it. And so you can have a couple filters there when you're working with coding variants. And this variant annotation package will help you with that. It will tell you like how many reads overlap each of your coding variants, and then you can apply a filter there, for example.
So Devika qualified the question, which was, are there detailed instructions to do the variant analysis in your own sample? I'm saying, I mean, when I run the speakeasy pipeline from
my own samples. Yeah, so if you run a speakeasy on your own samples and you don't have DNA genotyping data, what you'll be able to get is you'll be able to get this VCF file alone. So I'll go back to what I'm reading here. So you'll be able to get this merge variants.vcf file. Now, what can you do with it? If you only have the RNA infer genotyping information, right? These will depend on your study design. Imagine that you're studying, let's say, 10 donors, but you're generating data from, let's say, the lung and the brain for each of the 10 donors. So that means that you have 20 total RNA-seq samples, but you have the 10 donors. And so if you take into account that the variants for your 20 samples and you correlate them, you should be able to get small little groups where you have 2 samples that are very highly correlated with each other in the variants, and those should correspond to your 10 donors, those 10 groups of 2. If they don't, then that means that you might have a sample swap if the labels don't match, or you might also have had sample contamination or or maybe accidentally you duplicated one sample and didn't sequence another one. So all of those things can happen on the experimental side when you're preparing the data. We've had cases. We've had about like between a 2 and a 5% error rate in the data generation process across the samples that we generate at the Library Institute. So if you start to generate like thousands of samples, you start to see cases like that. That's why we wanted to look at the data itself to try to identify any sample swaps. If you have DNA genotyping data, then you can also try to match your RNA to the DNA. If you have RNA and DNA, you can check only among the RNA, only among the DNA, but also between the two, right? So that's the scenario that we're illustrating here on the speakeasy example, where we have all three sources. So you can check inside of RNA, inside of DNA, and between the two files. But if you only have RNA, then you can only do a portion of the analysis. So right now, what I'm only going to do is part of it. So of the different types of analysis that you can do. So we'll look here. What are here the call names of Geno Speakeasy? Let's explore this. So here, this is only the samples that we have. So we're going to use a bit of tidyr here just to separate the samples. Why we're doing that? That's because inside of those names we have actually information about what is each of the samples here. So this is the RNA ID for each of the samples, and we only want here the accepted portion of it. And so this has to do a little bit with like how our names are and in the data that we generated. And so this could be a little bit different for the data that you work with. Now, we're going to make all the 0, 0 genotypes as 0, the 0 ones as 1s, and the 1 ones as 2. Once we have that, then we have a genotype matrix that we can start to work with. We have a lot of NAs because a lot of the variants were not observed at enough quality in our data. But then using that information, we can compute the pairwise correlation across the samples. And so this is from correlation across across these coding variants that we just observed. And so once we have that correlation matrix, we can make it into a little heat map. So actually, let me actually go to the files panel under vignettes to open the code for that heat map, which I accidentally forgot to show. Yeah, so I accidentally forgot to set that to echo equals true. So here we're going to use this pmap function to make that little heat map. And so in this case, we only were supposed to have, where's the image? I don't know where I'll go back to the vignette. Here we can see this correlation matrix, and we don't expect to really like to have low correlations across the board because none of these samples were supposed to be duplicated. However, some of them are duplicated, and so we do see some high correlation values in the off diagonal. And so this was using the data from the RNA side. We can repeat the same process using the data from the DNA genotyping side, which if you have it available, you'll want to use, but you might not have, as we discussed with, sorry, I forgot your name, with Devika. Wait, I guess there was a follow-up question.
Yeah.
Does SPEAQeasy do also the DNA genotyping analysis? No, SPEAQeasy does not do the DNA genotyping analysis. So you have DNA genotyping by yourself internally in our institute. Like Josh Stolz has written some code for processing the DNA genotyping with the TopMed imputation panel using the Michigan server, I think. But that code is not part of SPEAQeasy, and we actually haven't shared it either. So, but that potentially could be another next little project, right? To write a pipeline for processing DNA genotyping information. I ignore if there are other pipelines out there already that do that. There might be some. Going back here, so we can repeat again the same process of throwing the DNA genotyping information into zeros, ones, or twos to compute a correlation matrix for the same variants, but those that are observed on the DNA genotyping side. Let me see here, what are the dimensions? So we have our 42 brains, and in this case we have 746 variants from the DNA genotyping side. So with those, we can make a second heat map. And so here we observe that on the DNA genotyping side, all of those brains are unique. They don't overlap. They don't have high correlations with other brains because only the values in the diagonal are high. So there are no issues right now on our DNA genotyping side. There's only some issues on our RNA. Cool. So let's continue with the vignette here.
Can I ask a question here, Leo? So we've got a case where maybe there's transplant cells and host cells. Will this be sufficient to discriminate the genotype of those two individuals so you can determine which is which in an RNA-seq experiment?
It could potentially help you do that. You might not have correlations of one, though. You might have a mixture, so there might be around 0.6 instead of around 1. We've seen these actually. Sometimes we have an off-diagonal correlation value here that is around 0.6, and that can happen if there was an accident in the library preparation and there was a little bit of contamination from one sample to the other. So I would imagine that in your case, when you're transplanting from one donor to a second donor, you might actually have on the RNA side data from donor cells and the transplant donor cells and the transplant recipient cells, right? So that's why you might have a little bit of a mix. I don't know if your protocol is very good and you only get cells from one or the other, then you'll be able to find who they are, right? But I suspect that you might get cells from both. So then you would get a mixed correlation. Does that make sense?
It does, yeah. Thank you.
Sure. So here, we don't have actually the same set of variants measured on the DNA and the RNA. In our small subset of 42 samples, maybe some of the variants that we use for our larger projects at the Libre Institute, we're not observing these 42 samples. So we're going to subset them to the common ones. And out of the 742 variants that we have here on the DNA level, we'll have 662 that are common between the two. So now that we have common variants, in the RNA and DNA, we can actually now compute a correlation between them. So let's compute that correlation again. And why do we want to do this? Right. So we're going to try to find what are the sample swaps. And so sometimes this can be easier to do like on a long data frame, just because then you can start to use like some of the tidyverse functions. So we'll make here a long data frame where we have, let me look at this object head of it. I'll close the source file. So what we have here is we have a specific here a sample on the rows. The samples on the rows were from the DNA genotyping, and then samples on the rows on the columns, which are from the RNA. And so here there are specific IDs for each of them. These IDs here are a bit more complicated, but those are the DNA IDs, and we'll be able to transfer those into an ID that is more of the brain number. But right now, we have two sets of IDs and a correlation between them. So this is where some of our other files come into play, such that we can add names. Did I miss a step? I feel like I missed a step because I'm not supposed to get NAs. I might. Yeah. Sorry, I missed a step on my code. Let me make this run this again. So 642. That correlation, convert to long format. Yeah, still an ace. So I probably missed it earlier. Sorry about that. I was trying to run the code interactively, and I forgot a line while talking.
Leo, given that there's 10 minutes left, maybe it depends on how things are going. Should we focus on just the HTML, or do people have additional questions or anything like that?
Yeah, sure. If people have any questions, I'll be happy to answer them. I was about to finish this this section on the DNA on swapping, which I thought is like I think is like interesting because it's different from what we see elsewhere. Okay, yeah. So these codes. Okay, I reran all the code. Sorry, I had missed a line there. So now I can. Now we have here the actual brain IDs. So we have here the brain IDs from the column. Those were the RNA IDs, and then from the rows, which were the DNA IDs. So we can see, for example, here, this first line is is the same brain on both sides, and we're only showing those that they're supposed to be the same brains, but the correlation is less than 0.8. So why is that a problem, right? So that's a problem because they're not matching to the brain they're supposed to match. And we can see here the correlation values are actually fairly low, except for this one that is 0.76. So that's one type of problem there that we can see once we're comparing the DNA versus the RNA. On the flip side here, we can also ask which are the brains that do not match. So here we see 2473 matching to 2260 on the ID labels, but they have a high correlation. So the data is telling you that they are actually the ones that they should be. So if you look at tables like this, you can find like patterns. And so, for example, here we have two brains, 2473, that match 2260, and vice versa. They have high correlation values. And also, 2473 doesn't match itself here, has a low correlation, over here. And so this is one of the simpler cases where we can be fairly confident that there was a label mismatch, and we can simply relabel these brains to be the other label, right? And vice versa. So we don't need to throw the data out. However, there's some other more complicated cases, like this 2275 brain, which doesn't match itself, even though it's supposed to, but we don't actually have any strong matches with any other brain. So at this case, this is not a problem that we can try to resolve, and so we need to drop that sample. So the next set of code here, what it will do is it will like drop that 2275 brain, and then it's going to fix the IDs for the 2260 and 2473 brains. Once we do that, our phenotype information also has additional information that was not provided to SPEAQeasy. And that's because this is very experiment specific. So in this case, we actually know information about what is the brain region, the race, the primary diagnosis, sex, and age at the time of death for these brains. So we'll add all of that into the column data of our range summarized experiment object at the end. And you can save it at this point because this will be the main object that you would use for your differential expression analysis. So in interest of time, I'll just mention here that the HTML explains a little bit of how you can actually do a differential expression analysis with these files. Now, if you're familiar with Lima or some of those other packages, then this section will be redundant with information you already know. But that's the point of SPEAQeasy, right? At some point, we have output files that are easy to analyze with any of the Bioconductor packages that are available out there for a differential expression, including like Lima, DC2, HR, and some other ones. And so this section of the HTML shows you how to do that type of analysis using packages such as Cluster Profiler, HR, Lima, and so this could be good to go through on your own after this workshop is done. And Josh and I will be around if you have any questions, or you can find us outside of this workshop. So what we show here is just the standard Lima analysis, where you can find some differential expressions, visualize them. You can make a little heat map, for example. The specific data that we're using here doesn't have a very strong signal, so you don't find dramatically different differential expressions. You only find here like 2 of them, for example. There's not that many in this example data set, and that's because the differences in expression between bipolar cases and control individuals are very subtle, and here we're only using 40 samples or so. Now, let me see if there are any questions in the meantime on the feed.
There aren't anything in the Q&A, but in the feed, there's been kind of a lot of excitement about sample swaps.
Yeah, sample swaps are they can be really hard to resolve, and so we've tried our best with Nick and Josh, and ultimately you can explain swaps multiple ways. So in our case, what we have to work with is we sometimes have more than one DNA genotyping assay for each donor brain, and so the 2 different DNA genotype should match to each other, right? But that's not always the case. And then we have multiple RNA-seq samples per donor, and some of them were done across the years. So you might see that some of them in the beginning maybe matched, and then they stop matching, right? And so you can have a lot of different sources of problems. One of them could be like maybe someone went to search for the brain, dissected it to generate the data that we needed for RNA-seq. But then once they put it back into the freezer, they put in the incorrect location, like maybe when the library preparation was being done, maybe there was a little bit of an issue with a pipette, and so you can get a little bit of contamination. So yeah, this can get really complex, and our goal is to try to avoid having to throw out data as much as possible, right? And we're able to recover. We have around 5,500 bulk RNA-seq samples at the Libre Institute. We're around 2,000 DNA genotyping measurements, and so we're able to recover some of those swap brains using this information generated from SPEAQeasy. I don't see any other questions, but
can I ask if how much of this pipeline could be applied to single cell data?
So when you're working with single cell data, let's say there's different types of it, but let's say you're working with 10x genomics single cell data, that tends to be 3' RNA-seq data. If you look at the 10x genomics website, for example, for their software called Cell Ranger for aligning, they have a recommendation there that you might be interested in aligning instead of to the mature mRNA, so mRNAs, you might be interested into aligning to the pre-mRNAs. And that is actually a common case for us when we're working with a single nucleus data instead of single cell. And all of this means that you might need to change what is the reference annotation that you're working with and how you're working with that annotation, right? So you might actually want to consider aligning to introns, not only exons, for example. That's one change. Another one is like maybe the speed of the software, and also how the FASTQ files are provided, because in single cell data, you typically use sequence tags, barcodes. You typically use barcodes to identify the different cells, and you might have to trim the FASTQ files to remove that. The short answer to all of that is that like speakeasy would need to be adapted quite a bit if you wanted to work with single cell data, and we currently don't recommend using it for single cell.
Thank you for a great workshop.
Thank you, everyone, and thanks, Nick, for presenting, and Josh for making the material.